Hello and welcome to StrandExplains, a podcast where we discuss what's happening in the world of precision medicine, from weird case studies and interesting facts to interviews with leaders in the industry. I'm Sakshi Shingal, and today I'm joined by my colleagues Sumedha Sudhaman and RK to talk about the feasibility of using genomic biomarkers in response to immunotherapies to treat pediatric cancers. Sumedha recently published a paper on the topic, which is said to be in Nature Medicine. She got her bachelor's and PhD from Delhi University in genetics, following which she held a variety of roles as a research scientist and bioinformatician before heading to Toronto to do her postdoc at Sick Kids. We are now lucky to have her as part of our team as a senior account manager for business development at Strand. Hi, Sumedha. It's so good to have you on the podcast. How are you doing today?
Very well. Good morning, Sakshi. How are you?
Good evening to you. I'm great, thanks. Yeah, so today's podcast, we might have a few audio technical glitches. We apologize for that. We're working on two different time zones. RK is currently situated in Kolkata. I'm in Bangalore, and Sumedha is in Vancouver, which has meant some, for me at least, and I think RK as well, our usual recording places are not available. So we apologize for any audio in the background. We're trying our best, but these things happen. So Sumedha, like I just mentioned, you're in Vancouver, and you and I actually have a shared history shared a location in that you were working in Toronto in Sick Kids, and I actually happened to be living in Toronto probably around the same time, although obviously we didn't meet. So what was that like working at a hospital which is crazily enough called Sick Kids?
Oh, it was incredible. And where I was working and the lab that I was a part of was part of the Arthur and Sonia Labatt Brain Tumor Association. And basically, it's a hospital for sick children. So therefore, it dealt in pediatric cancers and pediatric brain cancers, which are actually the worst of the lot because you can imagine like a child having a brain tumor is scary enough. So yeah, it was quite an amazing experience in terms of the work, but of course, it was, every step of the way, even as a researcher, you're always thinking about whatever you're doing is impacting these young children who are developing such aggressive forms of cancer.
Of course, it must be heavy. It must be, I'm sure, gratifying, but not always the easiest to do. And just working on such an important topic. At least I feel like it's such an important topic. It's incredible. So you recently released a paper on the use of genetic biomarkers of response to immunotherapies to treat pediatric cancers, and I thought that was so fascinating. And we thought it'd be a really great idea to have you ask you to come and talk to us about it. So maybe just taking a step back, Sumedha, before we jump into the meat of the paper, could you explain the role that DNA replication repair plays in cancers?
Of course, Sakshi, that's it's interesting that you mentioned that because that's actually, it's important to realize what it looks like without a defective system that comes into place that leads to the cancer. So in normal cells, what happens is in the course of DNA replication, there are some errors that are made that are fixed by certain protein complexes within the cell itself. One of these complexes is known as the mismatch repair system, and it consists of a group of proteins that are coded for by several genes. I'm going to talk about four main genes that are PMS2, MLH1, MSH2, and MSH6 that are a part of this, that are known as the mismatch repair system that pair the mismatch bases. And the other component is the DNA polymerase itself and its proofreading ability. And there are two genes that are basically responsible for the proteins that perform that function, and those are Pol E and Pol D1. So in a regular scenario, what would happen is as the cell undergoes DNA replication, these systems would come into place and they would come and check and proofread and correct the errors that are made in the course of normal replication. Now, what happens oftentimes is in certain populations where there are high rates of consanguinity or that tend to be endogamous, you do see that sometimes when an individual has a homozygous mutation or a compound heterozygous mutation, which means they have two defective copies of the genes of any one of the mismatch repair genes. In that situation, these individuals have something known as a genetic predisposition to develop these cancers because their mismatch repair machinery becomes faulty. You can imagine if any one of these proteins, these four proteins that I talked about, were defective or they didn't function, then the entire complex will not be able to do its job, which is to repair the errors that would have been made in the course of normal replication. So what happens as a result of this is that these genetically predisposed individuals that have these mutations keep accumulating random mutations all over the genome. So you can imagine over a period of time, based on several different cell divisions all over the body, these individuals and these children start developing some of the most aggressive forms of cancers that are known, which tend to have a high mutation burden because of the mechanism that I just explained. And these cancers tend to be what we call as hypermutant cancers, or cancers with more than 10 mutations per megabase. And these cancers that have already such a high mutation load, now you can imagine their mismatch repair system is still not functioning. And cancer is just uncontrolled cell division, right? So in a cancer situation, all the cells are dividing anyway uncontrollably because they're cancerous. They also have a defective mismatch repair system. So therefore, they're accumulating mutations at a very, very crazy rate, which results in an accumulation of a somatic mutation in one of the DNA polymerase proofreading domain genes, which I talked about, either POLE or POLD1. So imagine an individual that has a genetic predisposition that is mismatch repair deficient, or MMRD, also acquires a somatic hit in one of the polymerase proofreading genes. Now that individual doesn't even have the polymerase proofreading machinery to salvage whatever the mismatch repair machinery was trying to would have done otherwise, right, in a normally functioning cell. So therefore, this individual has a completely ablated replication repair system, or this person is completely replication repair deficient. So these cancers that are RRD or MMRD plus PPD, that is mismatch repair deficient and polymerase proofreading deficient, these cancers become ultra hypermutant. So they go from hypermutant, which is 10 mutations per megabase, to 100 or over 100 mutations per megabase, because their entire replication repair machinery is completely faulty and it doesn't work. So you can imagine why these children end up getting really aggressive cancers. And these cancers are not restricted to any one tissue of origin, because you can imagine they result from not a defined set of genes that are acquiring mutations, but all over the genome. So therefore, these cancers crop up all over the body, sometimes even at the same time, which are known as synchronous tumors. You can have an individual with a brain tumor as well as a GI cancer.
Wow, that's quite frightening, really. And like you're saying, it sounds like it. You said it's a very aggressive form of cancer with, from what I understand, rather abysmal survival rates. So, what are treatment options available for children with these types of cancers right now?
So typically, what would happen is these children that develop these aggressive cancers, when the first cancer is diagnosed, they would, their clinician would, based on the location, etc., they would try for either chemotherapy, radiotherapy, or surgery. But these cancers, because of the way they grow and they just keep cropping up, they have some of the poorest survival rates to standard chemotherapy and radiotherapy, which is why it's been a nightmare, like treating these cancers for clinicians all over the world. But one ray of hope in all of this was this breakthrough study that was published by the group that I worked with, a few years ago, where these absolutely brave, incredible clinician scientists, Eric Buffet, Uri Tabori, and others, what they did was they actually treated a couple of kids that had these aggressive recurrent brain tumors or glioblastomas. They treated them with immunotherapy, and they found this miraculous response to immunotherapy, and which was like it was groundbreaking, that work, and which actually sort of sowed the seed to this study, which is basically a registry study where one started thinking about why these children that responded so poorly to anything else have started responding to immunotherapy. And which brings us to the mechanism. I mean, and that was basically why we decided to do this study, because the group that I was a part of at Sick Kids, was part of the International Replication Repair Deficiency Consortium, which had patients from all over the world that had this condition. And out of those patients, the ones that were treated with immunotherapy were then, we did a registry study where we retrospectively looked at all of those tumors, and we tried to see whether we could identify biomarkers of response to immunotherapy, so we could understand at a genomic and a molecular level what is actually happening in the tumor in terms of response to immunotherapy.
Thanks, Sumedha. That was fascinating. And so, what are the ages of these children? And when they come in, I guess they come in with worried parents. Do they already have a diagnosis when they come in, or could you speak a little to how the diagnostic path looks for them, and how do you explain something as complex as mismatch repair, or any of this, right, to a parent who's probably just looking for answers on how to make their kid better?
Sure, RK. Yeah, typically there's, a couple of different scenarios in which, these parents would come in. Like I said, because there's a genetic predisposition to this, oftentimes, one of the parents would have had something known as Lynch syndrome, which results in mid to late onset.
Colon cancer.
Yeah, colorectal
Yeah.
cancer, because that results in
Yeah.
mutations in one of the mismatch repair genes. So that is what results in Lynch syndrome, and they have GI cancer. So typically, that is what a genetic counselor would tell parents of kids who develop, let's say, cancer or start to show cafe au lait spots and the usual diagnostic features. Even before that, like if a parent has had
What are those spots?
cafe au lait spots,
Uh, what spots?
cafe au lait
Oh, cafe au lait spots. I see. So they manifest cafe au lait spots on their skin,
on their
right?
skin.
They just look like coffee colored spots,
Yes,
essentially, right?
and very oftentimes it's confused with neurofibromatosis, and that is
Yeah.
generally how it starts in these children.
So do they have tumors too, these children, or no? These are all, like, some of them are blood cancers, right? So they don't have actually any tumors. They just have these spots and.
Yes, very few have hematologic malignancies, but largely over 60% of replication repair deficient tumors are brain or CNS tumors. And the others are extracranial solid tumors, about maybe 20% or so, or 30% are extracranial solid tumors. So GI, bladder.
I see. Sorry, you were saying. I think I interrupted you, Sumedha, but yeah, please continue.
Yeah, sorry. Just to pedal back. So if a parent has like Lynch syndrome already, you know that because they already have one allele mutated in any of the mismatch repair genes, so you know that that is something that they need to watch out for. They would have had genetic counseling, so they're already a little aware about like what their kid might have, and if their kid has any kind of cancer, immediately they get screened. The first thing that's done is the tumor is screened as well as the blood. So they look for germline markers or germline mismatch repair deficiency, and this is the clinically approved way even now that's done is by immunohistochemistry to check for germline deficiency as well as to check for the tumor MMRD status or tumor mismatch repair deficiency status. So this is that's one scenario, and the other is when it just comes out of nowhere and there is no history of cancer. Usually there would be, like I said, because it's a genetic predisposition, but then if there's nothing in the family, that can also happen. It's the first case, and then oftentimes, usually if a child has it, if they have siblings, that's their sort of the first tumor at diagnosis would be, let's say, the oldest child that had the tumor, and then any other siblings would then, the genetic counselor would advise them to get screened pretty much every year or so. And oftentimes, and if they do find that they're asymptomatic or they haven't developed the tumor yet, if they are mismatch repair deficient, germline mismatch repair deficient, you know that they will develop the tumor at some point. So they're always watching out for that. So imagine, I can't, I'm a parent of a 6-year-old, and I can't even imagine what it must be like for these parents to go through this. It's bad enough to be told your child has cancer, to be told that if a child hasn't developed cancer, that your child will definitely develop cancer, and not just any cancer, one of the most aggressive forms of cancer. And imagine like a brain tumor, a GI tumor at the same time. Yeah, that can be pretty awful. So when they come in with that, so what I'm trying to say is they come in with a little bit of genetic knowledge already. And if not, that's what the International Replication Repair Deficiency Consortium aims to do, where, there are a lot of like patient advocates, and there's a lot of material out there that's provided to them to try and understand, what this entails. The network of clinicians all over the world is very tightly knit for this community. And, because it's also rare, it's not very frequent. So, wherever it is that community sort of comes together and ensures that these children sort of get the best treatment options that is available to them within their resources.
Thank you. Thank you, Sumedha. Yeah.
Yeah, thank you so much for the explanation. And like you're saying, it does sound just frightening, especially from what I've understood. These children are pretty young when they come in with these symptoms, maybe like 3 or 4 years old, if I'm not mistaken. And please correct me if I'm wrong. So it must be a very, very difficult journey and just difficult situation to have to deal with. One of the things you mentioned was there are certain things that the genetic counselors look out for, or tell the parents to look out for. What are the genetic biomarkers that are known to be flagged in these situations? Would those be those 4 genes you mentioned earlier, plus the 2 from the polypda?
Yes, so those would be the genetic condition that you would screen for. So in terms of screening, you'd screen for any mutations in all of those genes, which is at the germline level. And once an individual has a tumor, then of course the tumor is checked for you know mismatch repair or replication repair deficiency with these genes. But in addition to that, if one has access or to exome sequencing or even like the foundation medicine panel, the tumor mutation burden is also another indication. If these tumors, because like I said, the mechanism involved in these tumors is that they have random mutations all over, and their rate of accumulating mutations is way higher than in other tumors. So therefore, these tumors will almost always be hypermutant. So they would have more than 10 mutations per megabase. So in terms of genetic indications or hints that tell you these tumors are what they are, that is what one looks at. But that is before the treatment options. Immunotherapy isn't the standard of care that's administered to these children, which is why we decided to do this study as well, because we know it works. Also because of the mechanistic aspect, because in individuals, so immunotherapy historically has been successful in tumors that have a high mutation rate, like in melanomas and some lung cancers. And you know that in those cancers, when there's a high mutation rate, the reason why immunotherapy is successful in those hot tumors, as they are known as, is because when there's a high mutation load, what happens is within the cancer cells, there's a tendency for more number of foreign peptides to be presented outside the MHC complex of the cancer cells, which then triggers, in a normal situation, it's a dance between the cancer cells and the immune cells, right, in the that are around the cancer cells. And typically a cancer's evasion mechanism is to sort of lock in to those and to pretend like they're normal cells to the immune cells. So the way immunotherapy works is that you have molecules that go and put it in a very, very layperson speak, that they go and try to nudge the immune cells and sort of inhibit the cancer cell to be able to disguise themselves. So therefore, that's how immunotherapy works, and therefore the immune cells can then attack the tumor cells because they recognize them as foreign. But the way they do that is because they recognize these foreign peptides that are very abnormal, which are produced by random mutations in tumor cells. And in these cancers, like in other cancers like melanoma, etc., where you have a high mutation load, what happens is the chances of more foreign peptides being translated and presented outside of the tumor cells is very high. So therefore, the chances of those tumors mounting a very strong immune response is also very high, and therefore it's the immune cells that sort of then attack the tumor cells, and therefore which results in the shrinkage of the tumor, etc. So in these cancers, like I said, they're very aggressive. They don't respond to any other kind of treatment, but they are prime candidates for immunotherapy, as was shown in that JCO paper by Eric Buffet and others at SickKids, where they showed that in those GBMs, those recurrent GBMs, where basically they'd lost all hope in those kids, when they were treated with immunotherapy, because those tumors were so hypermutant that they immediately, it worked, and the tumors completely shrunk. I don't know, if you have a chance, you can go look at that paper and you can see the MRIs, and it's literally like the tumor and no tumor. Right? That's the difference. Yeah. And that's because of this immune response, and therefore the number of neoantigens. So those peptides that I was talking about are basically candidate neoantigens. And, so the process is more mutations, more candidate neoantigens, the better the immune response, and therefore immunotherapy works, or good response to immunotherapy. But even within this response to immunotherapy, and which is what this paper was all about, is that there is a variability, like even amongst hypermutant tumors. It's not like all hypermutant or ultra hypermutant tumors will respond, which is why we decided to do the study to try and identify what might be some genomic and immunogenomic biomarkers of response to that immunotherapy. And we sort of try to characterize this group of hypermutant and ultra hypermutant tumors.
I see. That's honestly fascinating, and maybe I'm a bit of a biology nerd here, just but seeing the way all of this works and the mechanisms behind it. And thank you so much for explaining it so clearly, because I know that these things can sometimes get very nitty gritty, but that makes sense. And it's such a cool idea, for lack of a better word, to use immunotherapies to try and target these cancers, because from my understanding, and I think RK and I've spoken about this before, immunotherapies are definitely a burgeoning field of interest, trying to weaponize your immune system to help you fight off these things. So it is always just fascinating to learn about the different ways in which that can be done. So maybe could you just talk about what the results of your study was? What did you guys see? What were those biomarkers and neoantigens you found that maybe predisposed you to not predisposed, sorry, but were predictors of
Response.
response? Yeah, exactly. Thank you.
Yeah, of course. So, like I said, we started out with these tumors. So, we worked with 38 patients. And like I said, because some of the patients also had multiple tumors. So, we had a total of 45 tumors from 38 patients. And all of these tumors were treated with immunotherapy. And I'm going to talk about it as tumors being treated with immunotherapy and not patients, because, like I said, oftentimes we also had interesting cases where we had a patient with two different tumors, different sites. One tumor and both of them were treated with immunotherapy. One responded, the other didn't, right? So we wanted to try, and which is again one of the reasons why we wanted to do the study, where we tried to identify what were the underlying biomarkers that could probably predict why one of those tumors responded and the other didn't.
Sorry, just to cut in there for a second, but that kind of sounds like you're treating you're practically treating more than just 38 patients. If you're looking at each tumor as its own case, that it sounds like you're effectively treating maybe 650, 60 patients as tumors, if that makes sense. Which, wow, sorry, just that's crazy.
Yeah, I mean, it was definitely like a different way of looking at it, like at least in for clinicians and biologists alike. Even when we were doing our survival charts, we didn't do it by patient, we did it by tumor, which is definitely a different way of doing it. But of course, we had to do it by tumor because at the end of the day, that's what we're looking at. And we're studying tumor response, not patient response in general, like overall. So yeah, so when what we did was just want to briefly describe the kind of tools that we used to get to the conclusions that we made. It was a computational biology study, which means that we had access to exome sequencing data from 39 of these 45 tumors, and based on the exome sequencing, we characterized the tumors based on their total mutation burden, which we used. The metric that we used, actually, we did a bunch of different things, but the main metric that we used was total SNVs per megabase, because that is what is typically used, and we wanted to compare because TMB has been shown to be somewhat an okay like biomarker of response. Like sometimes it works, sometimes it doesn't. So we wanted to see, I mean, that was the most popular and widely studied biomarker that other groups had done, so we wanted to see what TMB looked like in this cohort, and we found that both mutation burden as measured by SNVs per megabase and total microsatellite indel load, both of these correlated very well with response to immunotherapy and overall survival in these tumors. And I have to add, all of these tumors were studied. That is, the analysis on all of these tumors was done at baseline or right before immunotherapy was administered. So that's how we did the biomarker categorization. And in addition to genomic markers, we also looked at the tumor immune microenvironment, and we looked at immune markers such as PDL1, again, a very well-known marker, also controversial, but yeah, we wanted to look at, to see whether PDL1 correlates with response to immunotherapy or not. And we also looked at CD8 and a bunch of other immune markers. And to summarize the results from that we found, putting our genomic and immunogenomic analysis together, we found that the best way to characterize this unique group of tumors in these replication repair deficient tumors, we saw that a combination, the best biomarker was actually a combination of both genomic and immunogenomic and immune markers together. So I would say an immunogenomic combination of biomarkers was actually showed us correlated the best with response to immunotherapy and overall survival. So that I would say was, one of the highlights of the study. And this, we also looked at a subset of these tumors that were CNS only, because I think I mentioned that over 60% of these tumors are brain tumors. And we wanted to see in brain tumors because especially before, like, if you take immunotherapy out of the equation, brain tumors actually fared the worst. Like, they had really bad survival. GI cancers, we know, respond reasonably better. So in these tumors, we wanted to see how brain tumors responded to immunotherapy, and we saw a remarkable, this is for the, I think, the first study in the world that has shown that brain tumors show a response of over 60% to immunotherapy. So which is again one of the most remarkable findings of the study. And we saw that the responses to immunotherapy were sustained over time as well, which, if you look at the paper, like, you can see it on the swimmer plot. And that was also another very encouraging finding that we identified in the course of the study. And lastly, another small aspect of this was, so in a lot of these tumors that are given immunotherapy or are treated with immunotherapy, we have another unique category of response to that immunotherapy, which we call as flare, or a flare response. So what that actually is that, so you have response to therapy, that is a positive response. Then you have progression, which is, a bad response. And then you have flare, which is considered to be progression, and clinicians usually stop treatment, because what happens is the tumor, right after you give the tumor immunotherapy, within the first 1 to 1.5 months, the tumor just increases in size at a rapid rate. So it looks like the tumor is increasing in size, and therefore the tumor is progressing. But actually, we wanted to try and understand what that flare really was, whether it really warrants, like, stopping therapy, because usually that's what happens. These individuals are taken off immunotherapy immediately, or treatment is stopped because they think that that's progression. We wanted to find out whether it was progression or pseudo progression. This has been an ongoing debate in this field for a really long time. And in order to address that, we thought, we are fortunate to have access to tumor samples that were collected for immunotherapy and also at time of flare in about two patients. And when we looked at these, and we studied this at the RNA, DNA, and protein level to try and see what it looks like in terms of the immune microenvironment, and try to see what is going on in these flare tumors. Is it actually progression, or is it an immune response to immunotherapy? And I'm happy to say that we were able to our results showed that flare was indeed an immune response to immunotherapy. And also, as part of the study, we had about 10 to 12 tumors that showed this flare response. Out of these 12 tumors, our clinicians were brave enough to continue treatment for 4 of these tumors. So 8 were treatment was stopped, as is the norm, and for 4 of them, treatment was continued. And what we found was those 4 tumors went on to respond to immunotherapy. That is, the tumor size increased, but then as treatment kept continuing, they reduced. So, which is why we felt that maybe flare would be an immune response. And therefore, when we went back and we did that, comparison of, we looked, so what we did was, we looked at the RNA level, we did something that is known as immune inference, or which is single state deconvolution, which is nothing but you do RNA sequencing on the tumors on the baseline and at time of flare in both the patients. And you basically use a signature gene set of immune cells, and you try to identify the proportions of the different immune cell types based on their gene expression signatures. So when we did that, we found that there was an increase in the CD8 T cell subpopulation at time of flare as compared to the baselines in both these patients. Next, what we did was we looked at the T cell receptor rearrangement profile at time of flare again in comparison to the baseline, and this is work done with. I was a joint postdoc at both SickKids and the Princess Margaret. So and my other PI from the Princess Margaret, Trevor Pugh, like his group basically does, they have this technology which allows for hybrid capture of the T cell receptor rearrangement profiling. And when we looked at the T cell receptor rearrangement profile, or the repertoire as such, of the T cell receptor alpha and beta at time of flare, we found that there was an increase in diversity and clonality at time of flare, which basically what that means is that it showed that there was indeed a non-specific immune response to immunotherapy at time of flare, which was our evidence number two to say that there is an immune response that's happening here at time of flare. And lastly, what another thing that we did was we had access to immunohistochemistry, which we stained for immune markers, and we found that there was an increase in CD8 staining at time of flare in comparison to the baseline. So all in all, when we put this together, we found that it was very interesting. We were able to at least lend some credence to the fact that flare might actually be pseudo progression or be an immune response to immunotherapy, which I really hope clinicians world over will find a little encouraging when they see flare in such tumors.
Yeah, I see. And it's just crazy to hear that this is something like this can happen. And I think one of the things you mentioned was the bravery involved by both the clinicians and scientists to, have conviction in themselves and their belief that flare response is pseudo progression versus progression. And I think this paper just generally speaks to the risks and bravery that can be involved when it comes to scientific research, especially when you're looking at pediatric cases where, not somehow, but it is just slightly more possible, and you want to be as careful as possible. What do you think was the most challenging part of the study for you personally? What did you like? Obviously, this is you, like you said, you're a mother yourself, and I'm sure there were points where it hit home. So what was the most challenging thing for you in this study?
I feel like it's just watching or working alongside. Of course, like I'm not gonna make a joke and say the bioinformatics. It was frustrating, but apart from all of that, no. But jokes aside, I really think like working so closely with these clinicians whose day to day, I sort of like got a glimpse into what they go through like every day, like dealing with these patients and what it really means. And it's sort of like, I mean, I know like all of us get into science like thinking we want to make a difference, and we want to contribute to understanding disease biology in some way. That's why I feel like all of us are in this field. But I feel this especially because it affects like children, and children with such aggressive cancers, and to see like them like this kind of work, like you see that all the granting agencies, all the clinical trialists, all the pharma companies, or all the patient advocates, and everyone who's involved in any way, right? And all the scientists and researchers, clinicians, everyone, right? And it's all of them together, and the patients, of course, that and the parents, the families of the patients that are willing to allow their um the tissue material to be a part of this research study, and watching all of that and to see how important that really is, like what a difference it can really make to inform clinical decisions and translational medicine down the line, and to see it like that, so up close like that, to me, I wouldn't say it was the hardest or the most challenging bit, but I'd like to flip that over and say that was definitely a very involved experience. And you can't detach the emotion from that. Like it's sort of at the end of the study, when it culminates in a good way, like you feel happy and you feel that this it feels very gratifying, but at the same time you're also taken very close to what can actually happen, because in a lot of these cases, like a lot of this is a retrospective study, and we're doing this to look at biomarkers, hopefully to contribute and improve the treatment path of like other children and other tumors, not these kids and tumors where it's sort of set in stone what might have happened. A lot of them have passed on. So just to that aspect of it, and just to stay so close to that. I mean, for us it's like multiple tumors, but then actually imagine it's one kid with like multiple tumors. Just being up close and personal with that.
No, definitely. Yeah, like I said, like these pediatric cancers, just working with them can't be easy, and it sounds like it was definitely gratifying. I mean, I think one of the things I know Arki and I try to do is kind of relate back to those patient stories and relate back why people do the science they do, right? I think it's really ironic that when we are trying to say something is impersonal or, not emotional, we say it's clinical, right? They had a clinical manner about them. Where really, in my eyes, the clinical aspect is the most human aspect of science, because that is where you're working with people. And I think this paper and your work just speaks to how interrelated that human aspect of science really can be with how we are as people, I guess. If that makes sense. But yeah, so thank you so much for taking the time out to speak to us. It was really fascinating to have this conversation with you, to learn about this paper firsthand, because, I did read the paper, but getting that insight into how these decisions were made, why they were made, and it was just fascinating. Thank you so much. Really appreciate it, Sumedha.
No worries, and it's my absolute pleasure to have spoken to you and RK about this. And yeah, and I'm glad that I could be a part of this and talk about my work.
Of course, thanks. Thanks, RK.
Thank you.